All right, so now what we are going to do is we are going to take a look at some of our biological molecules. So these are organic molecules that are important for life. Again, our biological molecules are carbohydrates, lipids, proteins, and nucleic acids. What we are going to talk about in this lecture are the first two. We're going to take a look at carbohydrates and lipids, and then in the next lecture we'll take a look at proteins and nucleic acids. So one thing to mention about all of these biological molecules are things called monomers and polymers. Now, monomers, the root of this word is mono, meaning one. So our monomers are going to be our building blocks for these different molecules. So the carbohydrates have monomers, and these are going to be the building blocks or the simple molecules that we will be building into our complex molecules, which are our polymers. Polymers has the word poly meaning many. So this is where we're going to take our one individual block, our monomer, and we're going to put lots and lots of them together to make polymers, poly meaning many. So this is where we get this complex molecule and our simple molecules. So we can take monomers, put them together to make polymers, right? That would be a building up reaction, which would be a what? Anabolic or catabolic? Right, anabolic reaction. And then we have our polymers, which we can then break down into monomers. So polymers then are complex molecules, breaking them down into our simple molecules are monomers, which would then be what? Anabolic or catabolic? Right, catabolic. And so we have monomers and polymers of all of these different biological molecules. And so we're going to talk about each of these monomers and polymers in each of these biological molecules. So we'll start with carbohydrates. So carbohydrates, the main function of carbohydrates is energy. Specifically, we're talking about short-term energy. So our carbohydrates are our short-term energy source. We talked about monomers and polymers. Our monomers for our carbohydrates are monosaccharides. So monosaccharide, mono meaning one, and saccharide, or saccha, meaning sugar. So these are simple sugars, or single sugars. Now these contain carbon, hydrogen, and oxygen. So remember, this is our shorthand, CHO, or carbohydrates, because they are composed of carbon, hydrogen, and oxygen. So if they are composed of carbon, hydrogen, and oxygen, do you think they are polar or nonpolar? Right, they're going to be polar because we have this oxygen that's going to be pulling electrons toward it, whether or not it's bonded to a carbon or a hydrogen. Good, so they're polar, which means they do or do not dissolve in water. Right, they do. They dissolve in water because water is polar. Our monosaccharides, or carbohydrates, are polar, which means that they will dissolve in water. So if we take a look at our monosaccharide structure, we have a short chain of carbons with one carbonyl. Remember what a carbonyl functional group is, and many hydroxyl groups. So I'm going to draw out kind of a simple monosaccharide chain structure. Okay, so this is a drawing style that I have not yet used, but when we're talking about chains of carbons, when we draw a chain like this, each point is considered a carbon. Now, these carbons can have, as we have seen in this image here, a hydroxyl group attached to them. And what that means, if we were to kind of look closer, is that we have the carbon, which is the point on the line here. And then we have our connections, right, to the other points, which would be other carbons. And then here we have a hydroxyl group. Now, this is a carbon, and as I've drawn it so far, there are only 3 bonds to the carbon, and we know carbon has 4 bonds. What's not being drawn in here is that there's a bond to a hydrogen as well, giving the 4th bond. And then we can see the same thing with this carbon here. You can see that if I were to, I'm going to kind of draw it over here, but if I were to draw this carbon up here, it's bonded to these other points in the drawing, the carbon, the bottom right and the bottom left. And then what's not drawn to it are the hydrogens that are here. Hydrogens, drawing all the hydrogens in a long carbon chain can be very distracting. It gets pretty messy. And so this right here is a simple form or a simple way to draw organic molecules. So again, each point here is considered a carbon, and what's not drawn are the hydrogens. And we can assume that if there is a bond that's missing, that it's actually a bond to a hydrogen. So this is our simple monosaccharide. This is kind of a general monosaccharide structure. And now the location, so you can see the carbonyl group over here, carbonyl group, and you can see lots of the hydroxyl groups in here. So the location of all of the carbonyl and the hydroxyl groups matters. So remember we talked about isomers in basic chemistry. So we have our isomers. Remember this is different structures. For the same molecule, for the same atoms. All right, so we have our basic structure here with carbons, hydrogens, oxygens, and then we have isomers. And then remember isomers, they're different structures, and so they have different properties. And so this is where we get that the location of those carbonyl groups and the hydroxyl groups matters. And that's because they function differently. Now, this is our chain form of a monosaccharide. So a monosaccharide is a simple sugar, but monosaccharides can form a more stable ring structure. So again, remember our carbon, those bonds in the carbon can rotate and they can move. And so if we take our basic chain structure and we turn that into our ring structure, we actually get something that looks like this. Give me a minute. We'll draw in our hydroxyl groups. You can see we've already drawn in the carbonyl group. Okay, so if we take our chain and we rotate things around a bit, you can see that this carbonyl group actually ended up right in here, bonded to a carbon. Any point in here is going to be a carbon. This is also a carbon. The carbon, 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 carbon. And then we have our different hydroxyl groups here, just as we saw in the long chain. And you can count the number of hydroxyl groups. You can see it's the same number of hydroxyl groups. You can actually count the carbons. It's the same number of carbons and oxygens as well. And so we can go from our chain, our long chain, to our ring structure. And so there's actually this kind of rotating again. So when it's put into water, it can rotate around. And what we have all of these hydroxyl groups out here that are going to be interacting with water, making this polar, right? So even though we have these carbon-hydrogen bonds in here, we have lots of oxygens in order to make it polar. And we know that sugar dissolves in water, and so this is because of these properties, because we have all of these oxygen-hydrogen bonds, these polar bonds. So let's talk about these different monosaccharides, because you do need to know the different monosaccharides. So the monosaccharides, number one, the most important monosaccharide is glucose. So glucose is our most important energy source. So our bodies like glucose. That's what it wants, is glucose. When we're talking about monosaccharides, when we're talking about these first three, we're talking about the same molecular formula, and that's why we talked about isomers. So the molecular formula that we're talking about for glucose and for the others is the C6H12O6. So this is our molecular formula for glucose, but it's also the molecular formula for the next monosaccharide that we'll talk about, which is galactose. Now, galactose then is an isomer of glucose. So remember, that means that it is also C6H12O6, but it's the oxygens and hydrogens and things are going to be oriented differently. So galactose is an isomer of glucose, and we find galactose in dairy products. So found in dairy products, and in some legumes. It's galactose, and then our third monosaccharide, again an isomer, is fructose. So fructose, again, is an isomer of glucose. So the same molecular formula, C6H12O6, just rearranged. And this is the sweet sugar found in fruits and honey. And you can remember that because it's fructose, is fruit. And galactose, if anybody knows about lactose intolerance, galactose is found in dairy products and some legumes. And then glucose is our most important energy source. We'll be talking lots and lots about glucose throughout the semester. So these are the three monosaccharides that all have the same molecular formula. They are isomers of each other. However, there is a fourth monosaccharide that you need to know, and this is ribose. Ribose is not an isomer of glucose. Its molecular formula is C5H10O5. So it's the only monosaccharide that is not a C6H12O6, not an isomer of glucose. And ribose is an important part of our nucleic acids. So RNA and DNA. So we actually have kind of two different types of ribose, and we'll talk more about this when we get to our nucleic acids, but we have our ribose sugar, and then we have something called deoxyribose. And we'll take a closer look at those when we get to our nucleic acids. But again, they have a different type of structure, so they're isomers. All right, so we have our monosaccharides. And remember, our monosaccharides are our monomers of carbohydrates. And that's what we're talking about. Now, if we take our monomers of carbohydrates, because remember, these are our building blocks. If we take our building blocks and we put them together, we get different types of molecules that you should understand. So if we take two monosaccharides and put them together, we get something called a disaccharide. Di meaning two. So we take two monosaccharides and we get our disaccharides. So our disaccharides are formed by a dehydration reaction between two monosaccharides. So remember our dehydration reaction, we remove water when we are putting these two reactants together. So remember, it removes water. Let's put that in there, removes water, and it requires energy. So if we were to kind of look at our rings, OH, OH, I'm going to add a hydrogen on here. This is an oxygen. And I'm going to draw the same molecule here, just kind of backwards. Well, you don't even need to do it backwards. We'll just... All right, so if we take our two monosaccharides, so this is a monosaccharide, and this is our monosaccharide, and through a dehydration reaction, all right, what we're going to do is we're going to take this OH and this H here, and then this is where we're going to get our water. All right, so remember dehydration reaction, when we remove the H2O, then this oxygen right here is going to bind up here to this carbon. And then what we're going to get, or actually, yeah, the oxygen is going to bind to this carbon because then we have an oxygen between the two molecules. And I'll just kind of draw this to indicate this molecule and to indicate this molecule. So you don't have to sit there and wait for me to draw all those things again. So then these are our two monosaccharides. And then we get our one disaccharide. So let's take a look at some of our disaccharides that we get from our dehydration reaction. So our first disaccharide that you need to know is lactose. So lactose is where we will take glucose and galactose. So if we take glucose and galactose, two of our monosaccharides, and put them together through a dehydration reaction, then what we get is lactose, lactose being a disaccharide. And this is the sugar that's found in milk. Dairy products. The other disaccharide that you need to know is sucrose. Sucrose is glucose plus fructose. And the reason that you need to know sucrose is that this is table sugar. So the kind of sugar, the kind of white sugar that you would add to different things, table sugar. So the two disaccharides you need to know are lactose, which is glucose plus galactose to make our disaccharide of lactose, which we find in dairy products, and glucose plus fructose making sucrose, which is our table sugar. So we have our different monosaccharides, and I do want to take just a moment to kind of draw the monosaccharides so that you know the difference, and you can see that they are isomers, but that they have the same, I'm just going to put CH2OH here. So down, down, up, down. This one right here is glucose. Then I'm going to draw galactose. So in this case, you can see that I'm going to change the direction of the hydroxyl groups, and we're not going to get into kind of orientation and organic chemistry in really big detail here, but that means that it's a different orientation, making it interact with things in a different way. So you can see the hydroxyl groups face down, down, and then up, and then down here. Here it's up, down, up, up. And this one is galactose. Then the third one is fructose. Well, that was a little bit. Now fructose, you can see, is quite different. CH2OH, CH2OH, an up hydroxyl and a down hydroxyl. So this is fructose. So then if we have our glucose and galactose together, then we get lactose. And if we have glucose and fructose together, then we get sucrose. So then, as we said, if we take our monosaccharides right here, and we want to turn them into a disaccharide, right? We want our monosaccharides to disaccharides. This was our dehydration reaction. So our dehydration reaction, which is what we have drawn over here. So we lose water in the process and we end up with our disaccharide. On the other hand, if we want to break down, if we want to take our disaccharide, our complex molecule, and break it down into monosaccharides, then this would be our hydrolysis reaction. So monosaccharides going to our disaccharide is our dehydration reaction. We lose water and we make a larger molecule, complex molecule. If we take our complex disaccharide and we want to break it down into monosaccharides, then we have our hydrolysis reaction. So remember, that's breaking it down, and then in this case, instead of releasing water, then we're going to use water, and then it also releases energy. So how is this done, hydrolysis done? Hydrolysis is done utilizing enzymes. So for the enzymes that we're talking about, if we take lactose and we want to break down lactose into glucose and galactose, the enzyme that we are going to use to do that is lactase. So when something has the ending ase on it, that indicates that it's an enzyme. And typically, the beginning part is an indicator of what that enzyme is working on. So lactase is the enzyme that breaks down lactose. Similarly, if we want to take sucrose and break it down into glucose and fructose, the enzyme that's going to do that is sucrase. So again, ACE. So sucrase breaks down sucrose into glucose and galactose. So if we're breaking down our disaccharides into our monosaccharides, we're using the enzymes lactase and sucrase. Now we also build up, right? So if we are going to continue building up, so we've taken our monosaccharides and we've looked at the different monosaccharides. If we go back here, so we have our monosaccharides, glucose, galactose, fructose, and ribose. Then we have our disaccharides. When we have our disaccharides, we have lactose and sucrose, which are composed of two monosaccharides. Then we have our polysaccharides. So polysaccharides are formed by dehydration reactions between lots of monosaccharides. So we're going to talk about the different types of polysaccharides that you need to be familiar with. So remember our monomer to polymer, right? So our monomers are monosaccharides, and then our polymers are polysaccharides. So poly meaning many, so there are many, and then saccharides meaning sugar, so many sugars. The first most important polysaccharide that you need to know is called glycogen. So glycogen is the main form of stored glucose in animals. In animal cells. So glycogen is many monosaccharides, all attached to each other in long chains. So our body is going to, as we eat food, we bring food in. So let's say we eat a potato, for example, we bring in starch from the potato, which is a carbohydrate, or we eat something sugary. Let's say we eat some ice cream or something, which has sugars in it, or let's say we eat just some candy or something super sugary, or soda. So we eat that. In our digestive tract, remember in our digestive system, we're going to take our polysaccharide, our polymer, and break it down into our monomers, which in this case for carbohydrates, we're taking down our long, big molecules of sugar, polysaccharides, and in the digestive system, we'll break them down into our monosaccharides. Then they get into the bloodstream. We'll talk more about this later on in the semester, but the glucose is all going to go to the cells of our body. So it gets into the bloodstream. The blood is going to carry the glucose all around in the body, looking for any place that needs energy. So trying to get glucose to all the cells of the body so they can use it to live, to function. Once all of the cells of the body are filled up with glucose, and we have an excess of glucose, we've eaten more sugar than all of the cells need, then that extra sugar, that extra glucose is going to go to the liver. Once it gets to the liver, it's going to be then turned into a polysaccharide of glycogen. And then it's going to be stored in the liver for times when there is not enough glucose in the blood. So if you eat a big meal that has lots of sugar in it, you eat it, the body breaks it down, we take that sugar, we put it to all of the cells of the body, then we have excess of the sugar, excess glucose. It's going to go to the liver. The liver is going to put it together, build it up into these long chains of glycogen, and then it's going to keep the glycogen stored in these liver cells. And then let's say you are going on for the rest of the day, it's been 3-4 hours now, you haven't eaten, then your body is going to go to the liver and say it's time for you to break down that glycogen, turn it into glucose so that we can get more energy. So then the liver is going to release that glycogen. It's going to break it down into glucose and get that glucose into the bloodstream to feed the rest of the cells of the body. So that's our first polysaccharide, is glycogen. The second polysaccharide you should know is starch. So starch is the main form of stored glucose in plant cells. So the main form of stored glucose in plants. So within their cells. So starch, we typically think of starchy things like potatoes. So potatoes are a good example of something that's very, very starchy. Of course, there's lots of other starchy things. There's lots of starch in breads. There's lots of starch in various different types of vegetables. Carrots have a lot of starch. But potatoes are really, really starchy. So looks just like the glycogen that we were talking about. So these long chains, we take a lot of glucose and we stick them in long chains together. It's a different kind of formation. We're not going to talk about that, but we have long chains of stored glucose in plant cells is starch, and it's a similar type of process. So plants are going to be making glucose, and when they make glucose, they go to all of the cells of the plant, and they give it glucose, and then the cells are going to store the extra glucose in the form of starch. The third polysaccharide that you should know is called cellulose. So cellulose are very long, straight chains of glucose. So while we said that starch is the main form of stored glucose in plants, meaning for energy, and glycogen is the main form of stored glucose in animals for energy, cellulose is a stored form of glucose in plants, but it's not stored for energy. What it is it's used to make the plant cell walls. So rather than being a stored energy product, instead it's a structure molecule. So it's a structure molecule to make plant cell walls. So in plants, they have a cell wall outside of their plasma membrane that makes them sturdy and makes them strong. And so this is a structure molecule. And cellulose is not digestible by humans. And typically what we do is we call this fiber. So when we eat plants and we talk about how it's a high fiber meal or we're eating lots of fiber, what we're talking about is actually eating cellulose. So this is what's giving the cell wall structure to the plant. And so these are straight chains, meaning that they don't have any branches on them. So the other two that we've already talked about, you notice that I made branches on them. In cellulose, they're very long, straight chains rather than branched chains. Now, the last polysaccharide I just want to mention really quickly is not as important for this class, is called chitin. And chitin is stored glucose, or not stored glucose, sorry. Let me erase that. We'll just say structure molecule in animals and fungi. So if we take a look at these really quickly, glycogen and starch are for energy storage. Energy storage in animals, in the case of glycogen, energy storage in plants, in the case of starch. Cellulose is a structural molecule, so a structure molecule that's important in cell walls of plants. Chitin, that's how that's said, chitin is a structure molecule in animals. And what we mean by that is it actually forms the exoskeleton. So the exoskeleton in various animals like insects. And then it's also found in fungi in their cell walls. So chitin we're not going to really focus on as much. I just wanted to mention that there's also a structural carbohydrate molecule in animals as well as plants. So we kind of have glycogen is for animals, and chitin is for animals, and starch is for plants, and cellulose is for plants. So that summarizes kind of what we're going to talk about for the carbohydrates. So you should know the different monosaccharides, so the building blocks of carbohydrates. You should know their function, that our carbohydrates are short-term energy storage. Our monomers are monosaccharides. You should know, sorry about that. You should know what the disaccharides are. You should know the enzymes related to the disaccharides, breaking those down, and then you should know the polysaccharides and the different functions of the polysaccharides. Now what I'd like to talk about are lipids. So our second biological molecule are the lipids. And for the lipids, our main function is long-term energy storage. Also forms membranes and important in communication. So let's take a look at the lipids. So just like our carbohydrates have a monomer, our lipids have monomers. Our monomer for lipids are called fatty acids or a sterile ring, and that depends on the polymer. So we'll talk about that in a moment. Our lipids have lots and lots of carbon-hydrogen bonds, and then only a few oxygens. So unlike carbohydrates where we had lots of the hydroxyl groups you saw in those individual molecules, we had lots of carbons, lots of hydrogens, and lots of oxygens. Our lipids are mostly made up of carbon-hydrogen bonds and only a few oxygen. So if we are mostly made up of carbon and hydrogen bonds, are lipids going to be polar or are they going to be nonpolar, do you think? Right, nonpolar, because remember, carbon-hydrogen bonds are not polar. So then, if they are nonpolar, are they going to be dissolving in water or not dissolving in water? Right, they're not soluble, or they do not dissolve soluble in water. Because remember, water is polar, and polar dissolves polar, nonpolar dissolves nonpolar. So they will not dissolve in water, meaning they are hydrophobic. hydrophobic, right? And our carbohydrates are hydrophilic. So there are three different types of lipids that we'll talk about. So we have triglycerides, we have phospholipids, and sterols. So let's take a look at our fatty acids. So we'll take a look at our monomers first, and then we'll build up to our polymers. So our monomers, the one that we're going to talk about first, that we see in both our triglycerides as well as our phospholipids, are fatty acids. And we have different types of fatty acids. The first type of fatty acid we're going to look at are saturated fatty acids. And this may sound somewhat familiar to you from nutritional labels on different food items. You can see that there's a label that has listed out saturated fatty acids. It has perhaps sometimes monosatcharides and polysaccharides. And so we're going to take a look at those. So first we have our saturated fatty acids. Saturated fatty acids, the reason they're called that is because they are completely saturated with hydrogens, meaning that they contain only single bonds. And those single bonds are all carbon-hydrogen bonds. So what we have are carbons attached to carbons, attached to carbons, attached to carbons, and all of these carbons have hydrogens. They are completely saturated, meaning completely filled up with hydrogens. So if the carbon is not bonded to the other carbons next to it, what it is bonded to are hydrogens. You can see it's completely covered in hydrogens. Another way to draw this is just simply like this, because remember, whenever we don't see a bond, then that's a hydrogen, and every point here, each of these points are carbons. So this right here is one representation of a fatty acid. This is another representation of fatty acid. That's the rest of the molecule we'll talk about. So a saturated fatty acid are those that are completely saturated, meaning they only have single bonds. So they have single bonds between the carbons. They have single bonds between the carbons and the hydrogens. Of course, hydrogens can only have a single bond. And so they are long, straight chains. They can rotate. The carbons can kind of turn around and twist and rotate, but there aren't any functional groups. So there aren't any functional groups attached to the long part of the fatty acid chain. There aren't any double or triple bonds. The other kind of fatty acid that we're going to talk about, or the next kind of fatty acid, is the unsaturated fatty acid. So unsaturated fatty acids are not completely saturated with hydrogens, which means they have one or more double bonds. So in this case, we could draw the chain form, and then there would be a double bond in here. Another way to draw that would be, of course, this. And then this would be hydrogens here. I don't want to spend the time to draw in all the hydrogens. But this is actually a little bit misleading as well, because this double bond in here, what this does is it actually puts a kink in the fatty acid. And so rather than it being a straight chain like we saw here with the saturated fatty acids, what we end up seeing instead is when we have our single bonds, they're nice and straight. And then when we have a double bond, it actually puts a kink in the bond, or a kink in the chain here. And then we have our singles and singles, and then we could have another double bond, and then singles and singles. And so what we end up seeing here is that we have this kink in the chain, where we did not see a kink in the chain of the saturated fatty acids. And the reason that this is important is actually why we see saturated fatty acids being or interacting differently than we see unsaturated fatty acids. What that means is that when we have a whole lot of saturated fatty acids, they can all lay right next to each other because they're nice and long and straight. So they're all laying really, really close to each other. And what that means is that at room temperature, these stay solid because they're forming a more solid structure. They're all very, very close to each other. They can get in really, really tight and close. And so some examples of this would be butter. So butter stays solid at room temperature, or lard, which is an animal fat. So these types of things stay solid at room temperature. Now, an unsaturated fatty acid has these kinks in them. And if they have these kinks, then what we see is they can't lay right next to each other. And what happens is we have these big spaces in here. And these big spaces, when we see these types of big spaces in chemistry or in living things, in molecules, these spaces mean that there's room for movement. So we see kind of wiggling around and movement. So at room temp, these things are going to be liquid. So some examples would be oils. So our different types of oils are the lipids that are going to be liquid at room temperature. So if you took some canola oil, vegetable oil, and you put them in the refrigerator, put them in the freezer, they may slow down their movement. They may have less space between them, which would make them more solid, but at room temperature, they're going to be liquid. Now, an example to that, as far as oil is concerned, is coconut oil. So coconut oil has lots of saturated fatty acids in it, and that makes it more solid at room temperature. It's not quite as solid as butter or lard, but it ends up being in the saturated fatty acid category. And saturated fatty acids are actually not as good for you. They're less healthy than unsaturated fatty acids, and that's because of this ability for them to lay right next to each other. So since they pack right next to each other, the saturated fatty acids, they can do that very same thing inside of our arteries, inside of the body. So they can stick very, very close to each other, and then when they do that, they can start to form plaques, and they can start to build up, and they can be really very difficult to kind of scrape off and get rid of out of the body. On the other hand, if we have a high amount of unsaturated fatty acids floating around in the bloodstream, then they are less likely to stick to the arteries because they have these kinks in them that makes them more liquid rather than more solid. And so then they are more healthy. Our unsaturated fatty acids are actually an essential part of our diet. We're not able to make all of the unsaturated fatty acids, and they're actually important building blocks in our body. We actually need unsaturated fatty acids in order for different things to function. However, our body is very easily able to make saturated fatty acids since it's just a long chain of carbons and hydrogens, so we don't need to get a bunch of saturated fatty acids in our diet. Now, I do want to mention really quickly a third type of fatty acid called the trans fatty acid. And you've probably heard of trans fats, as they've now been banned in various items. So trans fatty acids are unsaturated fatty acids with a double bond in a different shape. So what this means, without getting into a lot of chemistry, is when you have double bonds, they can be in such a formation that's called cis, or they can be in a formation called trans. And so what we see are the unsaturated fatty acids that I had already drawn. I guess that's not exactly how that would form. Hang on. There we go. So this formation here would be the cis formation. And these are the unsaturated fatty acids that we were just talking about. This formation would be the trans formation. And the trans formation actually is naturally occurring. In small amounts, however. So in small amounts, in meat and in dairy, so in animal products. And that's because what we're doing is we're taking those trans fatty acids that are actually in the meat of an animal and actually in the dairy and in the mammary glands of that animal. So they are actually naturally occurring in those things, but they're not naturally occurring in any other product, or in any other plant item, for example. Now they are produced, and the reason they're banned is because they're produced in large amounts by hydrogenation in manufactured food. So what they have done is that they have taken this cis formation of unsaturated fatty acids, right? We have our unsaturated fatty acids that have all these kinks in them, and they give space, and so they're fluid. But unsaturated fatty acids are not stable at room temperature. We said that they're oily and they're liquidy at room temperature. What that means is that they are not shelf stable, which means that they go rancid or they'll go bad if they're sitting on the shelf without refrigeration. So if manufacturers were to make things like crackers and cookies and things like that that are sitting on the shelf in the grocery store for long periods of time, they would go bad. They would go rancid very, very quickly. Just like if you made fresh things at home, perhaps you know that if you make fresh cookies or something like that, they only last a couple of days versus food products that are put on the shelf for years, even at a time. But what food manufacturers have done is they have taken this cis formation, they've put it in a container, and they've bombarded it with hydrogens. This is why it's called hydrogenation. So they take hydrogens and they add lots of hydrogens to it. They take the unsaturated fatty acids that are nice and kinked and have spaces and are more liquid, and when they hit it with a whole bunch of hydrogens, what it does is it changes it into the transformation. And this transformation, you can see, is actually quite straight. Since it is quite straight, it actually packs just like saturated fatty acids can pack right next to each other, which means that it ends up being shelf stable. Since it's shelf stable, that means it can stay on the shelf for years and years, and the manufacturers don't have to worry about food going rancid after just a couple of days. So they're straight due to the shape of this double bond in here, the trans version. And so they act more like saturated fats, but they're even less healthy than saturated fats. So they're less healthy because our body doesn't naturally make trans fatty acids. So our body kind of treats it like some sort of weird toxic chemical that it doesn't really know what to do with. And since it doesn't really know what to do with it, it's not really able to figure out how to break it down, or it takes a long time to break it down, we end up having these trans fatty acids floating around in our bloodstream a lot longer than we have unsaturated fatty acids or saturated fatty acids. And since that's the case, what ends up happening is that this can lead to higher amounts of atherosclerosis. It can lead to lots of buildup of fat and plaque in the walls of our arteries. And that's why trans fatty acids have now been banned from many things, because they are so bad for our health. So the healthiest fat are those that contain unsaturated fatty acids, and we see those in different types of oils, and those all come from plants, with the exception of coconut oil. The second healthiest, so slightly less healthy, or quite a bit less healthy, is the saturated fatty acids. So we're talking about butter and lard and even coconut oil. And then the least healthy are the trans fatty acids. Now, getting away from the health aspect of them, remember when we're talking about fatty acids, what we're talking about are the monomers of lipids. So now that we've talked about the monomers of lipids, let's take a look at the polymers of lipids, these particular polymers that work with these particular monomers, rather. So the polymer of lipids, so the first polymer that you should know are the triglycerides. So triglycerides are our long-term energy storage molecules. So remember, we already talked about carbohydrates as being our short-term energy molecule, our short-term energy storage molecule. So we have glycogen that's stored in our livers, And in our liver, we have all of this glycogen, and when our body needs glucose and we haven't eaten it recently, then our liver will break down the glycogen into glucose and then release it into the body. So let's say you wake up in the morning, you eat a big breakfast with some orange juice and some jelly and stuff, things that have sugar, things that have carbohydrates, some toast, and then you have that with butter on it as well. So you've gotten both carbohydrates and you've also gotten some fatty acids, some fat. So you take that into the body, you've eaten more carbohydrates than your body needs at the moment, you've eaten more fat than your body needs at the moment. So your body's going to store these things. So first it's going to store the glucose as glycogen in the liver. The fats are then going to be stored as triglycerides in fatty tissue, so kind of fat deposits in the body. So then when you have finished eating and all of these things have been stored, they've gone through the bloodstream, they've gone to the various places, they've been stored up. Then, say it's been a long day, you've been up, you've been doing things, it's been several hours later, you haven't eaten. Your body then is going to tell the liver to start breaking down glycogen in order to get glucose. Then, after it goes through and it uses up all the glycogen, it's breaking it down into glucose and getting that glucose into the bloodstream, then it runs out of glycogen. So that's our short-term energy storage. It breaks down the glycogen first and uses it as glucose. Then, if, say, you're not eating for the whole rest of the day, you haven't had a time to take a break and get some food, it's nighttime now, and now you're at the point where you've used up all of your glycogen. Now the body's going to go to your triglyceride storage. So it's going to go to the fat storage in your fat deposits. So this is why our triglycerides are long-term energy storage, is because it's after we've used up our glycogen that then the body's going to start tapping into these glycerides that are stored in the fatty tissue. So our triglycerides actually contain twice as much energy per gram as carbohydrates and proteins. So it's actually a little bit more than that. Our carbohydrates give us about 4 calories, which are quantities of energy, or ATP. So carbohydrates are going to give us about 4 calories, whereas fats are going to give us about 9. So I'll just kind of put the carbohydrates in parentheses since I didn't mention it before. So kilocalories are the technical term, but it's 4 calories per gram for carbohydrates, whereas lipids or triglycerides are 9 calories or kilocalories per gram. This is for our lipids. And when we're talking about lipids in this case, we're talking specifically about triglycerides. So now let's take a look at the structure of a triglyceride. So the structure of the triglyceride, so the polymer of our lipids, is a glycerol backbone. I'll actually write that in there. So the glycerol backbone plus up to 3, in this case, since it's triglycerides, there are 3 fatty acids. So the name triglyceride, so it's tri for the 3 fatty acids, and then glyceride is for the glycerol backbone in this case. So what we have is this glycerol backbone, which is a 3-carbon chain with hydroxyl groups. And we are going to join that via dehydration reactions to our fatty acids. Now, the reason they're called fatty acids is because we have our carboxyl group here on the end. And then we have our long carbon hydrogen chain. And then we have another one. I'm not going to fill in all the hydrogens. In fact, I think I'll just do the simple version here. And then the last one. Okay, so then you can see here that if we do our dehydration reaction, remember dehydration is when we're going to be combining things. So we'll lose water in the process of taking our monomers and putting them together to make polymers. So we take H2O here, and H2O here. So we lose the water there, and then these things are combined. So we have our glycerol backbone here, and then our 3 fatty acids. and that makes up our triglyceride. Now, the 3 fatty acids can be saturated fatty acids, as we talked about, so they're completely saturated with hydrogens, or they can be unsaturated fatty acids. And what that means is that each of these fatty acids can be any of those things. So one of them could be a fatty acid, another could be unsaturated fatty acids, and they can combine in various ways. So this is our long-term energy storage. That's the first polymer of the lipids that we want to talk about. So we talked about our monomer of lipids, which are fatty acids, so saturated fatty acids, unsaturated fatty acids, and then we have our trans fatty acids. And then we're talking about our polymers. So our triglycerides is the first polymer. Now the second polymer that we have for our for our lipids are called phospholipids. And phospholipids are lipids that are going to be forming the plasma membrane of a cell. And we're going to talk more about that when we get to the cell section. But the function of phospholipids are to form plasma membranes. And we're talking about cells here. But it's also in organelles in cells. So it makes up the plasma membrane, the whole outside of the cell, which is the barrier between the exterior. And then it also is part of the membranous organelles within a cell. So a phospholipid, the structure of a phospholipid is that we have a glycerol backbone. So again, we have our glycerol backbone, plus, in this case, 2 fatty acids. So remember, the triglyceride was 3 because of triglyceride, so the 3 fatty acids with a glycerol backbone. And now when we take a look at a phospholipid, we actually have a glycerol with 2 fatty acids. And then along with these things, the glycerol and the 2 fatty acids is where we get the name from. We have a phosphate-linked head group. So we have a phosphate linked head group. So this one typically is drawn with the head here, and then we have our 2 fatty acid tails here. So this is kind of our head, which includes our phosphate-linked head group and the glycerol backbone, and then these, of course, are quickly drawn fatty acid tails. And there are 2 of them. So this structure here, this is the head, and then of course these are the tails. And these phospholipids are amphipathic. So amphipathic means that this is a molecule, so any molecule that's amphipathic, including phospholipids, means that they are polar, not at, but polar on one end and nonpolar on the other. So if we take a look at our phospholipid, if we're talking about our glycerol, remember the glycerol had those hydroxyl groups on them. And then if we're talking about a phosphate group, remember our phosphate group had that phosphate, and then we had lots of oxygens. I'm not going to go through the whole thing, but lots of oxygens on it and hydroxyl groups. And so that was also very polar. So this region, the head region, is the polar region. And then the tails, as we know, are nonpolar. And so that's what makes our phospholipids amphipathic, that we have this head region that's polar, and since it's polar, it's going to interact with water. So remember, that means it's hydrophilic. And then we have these tails, which are nonpolar, which make it hydrophobic. And so then, and what ends up happening is that the head region is going to interact with water while the tails are going to be sequestered and move away from the water. So if we take a whole bunch of phospholipids and put them into water, which happens because our bodies are made up of large amounts of water, what they end up doing is they end up forming a bilayer. And so the heads face out toward the water, out meaning outside of the cell, but also in toward the cell, because in both cases, we have water on the outside of the cell, which is the extracellular fluid on the outside, and we have intracellular fluid, meaning on the inside. And then we have the tails face in. So when I say out, we're talking about outside of the bilayer, and when we say in, we're talking about inside of the bilayer, so away from water. So I'll do a very quick drawing of what that looks like. So we have our heads here with our phospholipid tails, or our fatty acid tails. And then they end up forming a bilayer. So those aren't close enough together, but then we have our phospholipids on this side with the tails facing toward the other tails. And so then this will keep going on this way and this way and this way and this way until they form a membrane around the entire cell, or if we're talking about an organelle, it will make the organelle. So on the outside here, this is where we have water on the outside of the cell, which is interacting with our polar heads of the phospholipids. On this side, then, we have water as well, which is interacting with the polar heads of the phospholipids. In here, however, we have our hydrophobic region. So there's no water in this area. This is our fatty region because remember, they are hydrophobic, meaning they like to stay away from water. They're water-fearing. And so this is what our phospholipids do if placed in water. They're going to form this bilayer. Now they can form a bilayer in this type of orientation where they're much larger, or oftentimes you'll see it kind of in a circular form, which is called a micelle. And so the phospholipid tails will all go interior. I'm just going to draw them straight really quick. Again, because we have water on the outside here, and then this is all fatty area on the inside. All right, so then we have our polymers, again, triglycerides and phospholipids, and we're going to look at our last lipid polymer. So our last lipid polymer, called the sterols. Now the sterols, their function is for structure and communication. The structure is 4 connected rings of carbons and hydrogens, and then most of them are synthesized from cholesterol. All right, so our sterols are, again, going to be lipids, and one of our most important sterols are cholesterol. So they're used for structure. We'll see that in the plasma membrane, and then they're also used for communication because a lot of our hormones are sterol hormones. So the structure are 4 connected rings, carbon-hydrogen rings, and then most of these sterols are going to be made from the base of cholesterol. So we take these sterol rings from cholesterol and we add certain things to them to make other things. So cholesterol is important because it keeps our plasma membrane of the cell fluid. And we're going to talk more about this when we get to the cells unit, but the cholesterol acts as kind of a big rock in the middle of the plasma, or in the middle of those phospholipids. The plasma membrane is constantly moving around, and so all of these phospholipids are kind of moving around, and the cholesterol helps to keep those fluid because they make kind of big spaces, so they're not packed in as close to each other, kind of like we mentioned with saturated fats, where if we take saturated fatty acids and we put them all next to each other, there's not a lot of movement. But if we took a big chunk of cholesterol and stuck it in there, then there would be more movement because there would be more space. So the cholesterol does that for the plasma membrane. However, if we have excess cholesterol in the blood, then it can increase the risk for cardiovascular disease. So if we have high amounts of cholesterol, then we increase risk for cardiovascular disease. So it's very, very important that we get cholesterol in our diet because it's very important for all of our plasma membranes, so every single cell in the body. But it's also important for hormonal communication. So if we don't get enough cholesterol, then we're not able to make the different hormones that make the body function. And that's the communication piece. However, if we get too much cholesterol, then in the bloodstream, it's going to build up, and it can increase the risk of it getting stuck into the inside of our blood vessels and then can cause serious problems with blood flow. And actually, our liver can synthesize cholesterol, so we don't even really need that much in our diet at all. So liver synthesizes cholesterol. So we can decrease the cholesterol we get in the diet because we can make it on our own. So cholesterol is one sterol that keeps the plasma membrane fluid. That's important. And as I've been mentioning already, we have our sterol hormones as well. So our sterol hormones are going to be the communication function. So communication between different parts of the body. So some examples, stress hormone, cortisol. That is a sterol hormone. And then also the sex hormones. So what we're talking about there is testosterone and estradiol, also known as estrogen. They're actually a group of hormones called estrogens, and that includes estradiol. Another type of sterol that's very important. So when we're talking about the sterols, we have cholesterol as a sterol. 2, we have our sterol hormones. And then 3, we have our lipid vitamins. And these are also sterols. So our lipid vitamins are A, D, E, and K. Those are our lipid-soluble vitamins. I'm not going to go into drawing them all out, but A, D, E, and K are all lipid-soluble, and actually that's what makes them a little bit dangerous as well. Just as a side note, you can take lots and lots and lots of vitamins that are water-soluble. So all the other vitamins other than A, D, E, and K are water-soluble. If you take an excess amount of these, our body will simply urinate them out. Because they're water soluble, they'll stay in the bloodstream and then they'll get filtered out in the kidneys and then they'll get urinated out of the body when the body has enough. However, if we're talking about our fat-soluble vitamins, our lipid vitamins A, D, E, and K, those are stored in our fatty deposits. So if you take lots and lots of A or D or E or K, then that can get stored up in the fatty tissue. Now, that can be a good thing because when we break down our fatty tissue, then we can get these vitamins into the bloodstream, but it can also be a very bad thing because if you take high levels of these vitamins, they are not urinated out. They're put into all of the fatty tissues. And then if you get to a point where you're breaking down your fatty tissues, say you are going into your long-term energy storage and you start breaking down the fatty tissue, or let's say you start to exercise a lot more and then your body's starting to break down the fat deposits, then you can start to overwhelm your body with A, D, E, and K, and it can start to cause some serious issues depending on the type of vitamin. So those are ones that you have to be concerned about taking too many vitamins. So that is going to end our lipids portion of this, and then the next lecture we're going to move into our proteins and nucleic acids. So just to summarize, when we're talking about our lipids, remember we have our monomers, which are fatty acids, or our sterol rings. We have our different types of fatty acids. Then we have our polymers, the triglycerides, phospholipids, and sterols. And then previous to that, we talked about our carbohydrates. So we talked about our monomers, which are glucose, galactose, fructose, and ribose. Then we talked about disaccharides, which was the combination of 2 of those. And you should know lactose and sucrose and their enzymes. And then we talked about the polysaccharides, so glycogen, starch, cellulose, and chitin. All right, so that ends this portion, our carbohydrates and lipids. In the next lecture, we'll cover our proteins and our nucleic acids. Monosaccharides.